Hello and welcome to our chapter on viruses. So very important things to remember or understand from this chapter, and we're really not covering the entire chapter, but I would like you to understand the basic structure of a virus, also distinguishing, which we will talk about in this video, between something called the lytic and the lysogenic reproductive cycles of a virus, and then understanding this basic idea of viruses as parasites and really requiring other organisms to replicate. They are obligate, intracellular, so they function on the inside of cells to really take over the machinery of the cell and replicate more viruses, but they can't do it on their own. They have to have another organism to do that. And then finally, looking at the life cycle or the reproductive cycle of viruses that infect animals and how they evade the the immune system of those animals while they're replicating and producing more of themselves. So, beautiful. I love this picture. This is an electron micrograph of a bacterial cell that is being attacked by these lovely little yellow viruses. Okay, so we're going to see this in action in the lytic versus lysogenic cycle, the virus actually attaching to the cell membrane of the bacterial cell and injecting its DNA to take over the cell machinery. Okay, so there are two ways this can go. And one way it could go is something called the lytic cycle. And in the lytic cycle, what happens is, we'll start up here, the virus injects its DNA, shown here in blue, into the bacterial cell. And just first, before we start off, these are phage DNA. A phage, a bacteriophage in particular, is a virus for bacterial cells only, bacteriophage. They are attacking bacterial cells specifically. And they inject the DNA, and one of two things can happen. It can go lytic, and it can stay really in its own, a little circle of DNA, and cause that cell to replicate more of its DNA, more of its proteins, to form more of its viruses, until the inside of that prokaryotic cell reaches a critical mass of virus molecules and causing that bacterial cell to lyse or to break. And then the phages go on to infect other bacterial cells. Okay, so that's lytic. And usually the lytic cycle occurs when the bacterial cell isn't in such a great environment. So the virus will take advantage of the bacterial cell while the bacterial cell is still alive to replicate its genome, shown here in blue, and its proteins, and kind of get out of there as fast as it can before that bacterial cell dies on its own in its environment. Now, if the bacterial cell's in a good environment, good nutrients, good temperature, bacterial cell's doing well, the phage DNA may do something a little different and go into the lysogenic cycle, where the actual viral DNA or viral genome, whether it be DNA or RNA, incorporates into the virus, incorporates into the prokaryotic genome. When it incorporates in, it's called a prophage. So it actually becomes part of that circle of DNA in the prokaryote. All right, so it will go through its normal replication process, the prokaryotic binary fission, and it'll replicate that viral genome. Now, all right, so if it's, again, in a good environment, it can go lysogenic, and it'll replicate the viral genome and have that prophage, which is, again, that viral genome in its DNA for generations and generations. And then maybe a day will come where the bacterial cell's in a bad environment. The phage DNA recircularizes, comes out of the genome, and it can go lytic. Okay, so one of two ways. And again, lytic, usually when the bacterial cell is not in such a great environment, it'll go lytic cycle for the viral replication. And if the bacterial cell is in a good environment, it also might just depend on the phage DNA or the phage type itself. It could go lysogenic as well. All right, now this last part here, I'm just going to briefly talk about animal viruses and how they evade the immune system. So here we have an HIV viral molecule going into a white blood cell of an animal and wreaks its havoc inside of the white blood cell, and it actually exits, or many of these HIV molecules will exit the white blood cell, but they'll take with them the white blood cell membrane. So it allows that HIV virus, and other viruses that infect animal cells do the same, to evade the immune system, because the immune system sees that virus encased in it doesn't see the virus, it just sees the casing, which is self or itself, and doesn't suspect at first that anything is amiss, and it won't destroy or target those membrane encased molecules right away. All right, so that is the overview that we are going to discuss here. If you have any questions, please let me know.